Hello everyone, my name is Shayan Nikakhtar, and in this video I'm going to give you a presentation on the cell. So in the previous video I mentioned that in the biological hierarchy, cell is that point where we are able to see the properties of life such as respiration, reproduction, uh, digestion. So these kind of properties of which represents life we are able to see at the point in the biological hierarchy. So we go from atoms to macromolecules, then to organelles, and from organelles we are able to reach this point, cell, which represents life. So if you look at a little bit of history, so the cell was discovered by Robert Hooke, and when he discovered it, when he was observing a piece of food under the microscope, he saw small rooms or small compartments. So small compartments. So that's what the word, and he named them cella, basically, which ultimately it came to be known as the compartment, which ultimately came to be known as the cell. So there are two basic main divisions of the cell. There are basically two main divisions of the cell, and one is the one is called the eukaryotic cell, and the other one is called a prokaryotic cell. Eukaryotic cell and prokaryotic cell. So the main difference between these two cells is that in eukaryotic, basically the word eu means true from Greek, and karyon means nucleus. It means true nucleus. And pro means false or not true, and karyon means nucleus. So this is called true. This basically word means true nucleus, and this word means like not true or false nucleus. And why is it called a true nucleus and why is it called a false nucleus? Because if we observe a eukaryotic cell, we will find that around the place where the genetic material of the cell is present, so around it there is a membrane. So the genetic material is enclosed by a membrane, so we say that this cell possesses a true nucleus, or it is a eukaryotic cell. Whereas if we look at the structure of a prokaryotic cell, so the genetic material is not enclosed by a membrane, and the genetic material is just concentrated in a location, and so the place where the genetic material is located is called a nucleoid. Nucleoid. It's an E right there. Anyways, so this is the basically main division. So you might think, okay, what kind of cells are called prokaryotic cells? Well, all the bacteria or archaea, which are called ancient bacteria, are prokaryotes, and all the other organisms, including you, me, and even unicellular organisms such as paramecium, they are all eukaryotic organisms because they possess a true nucleus. Anyways, the first part, which I just accidentally removed, was the nucleus right over here. So in the introduction of a cell, I just told you a little bit about the nucleus, that the eukaryotic cell possesses a true nucleus, whereas the prokaryotic cell does not possess a true nucleus. Now, so I've selected these organelles such as the nucleus, the endoplasmic reticulum, the ribosomes, the golgibodies, because basically I've divided the video into a couple of sections, and in this section I'm going to discuss about the organelles, which are mainly related to the producing or manufacturing functions. So the first is called the nucleus. So as I mentioned before, a nucleus of a cell is enclosed by a membrane. So this is, I'm drawing a nucleus right here. So this is the membrane, and it is called the nuclear membrane. Nuclear membrane. And the nuclear membrane has some holes or pores which are called the nuclear pores. Nuclear pores. And inside the nucleus, all the genetic material of the cell is concentrated, is located in a chromatin form. And this ball, you might say, which I'm creating right here, which I'm drawing, is called the nucleolus. So I use two words right here, which you might be thinking it's a little bit confusing, which is called, first is called the chromatin, and the other one is called, and another one which I used was the nucleolus. Nucleolus. So the chromatin form is basically what it is. What chromatin is it just it's just DNA plus proteins. So you know, like what so you might know what DNA is. DNA is basically the heredity material. It's in this form, it's in double helix form. And so what chromatin basically is just, it's DNA wrapped around. So you might be thinking a DNA is a long strain, like long piece of thread, which is wrapped around proteins. Like if I would just zoom out of this is just a zoom in portion, this is the zoom out portion, it would be like this whole DNA is wrapped around special kinds of proteins, which gives its particular shape and structure. And these proteins which it's wrapped around are known as histones. So these proteins are basically histones, which you may view them as structural proteins. Histones. That's an S right there. Histones. And this changes shape when the cell wants to divide by mitosis and meiosis. And you might encounter this term when you're studying mitosis and meiosis, is that when chromatin changes its shape into chromosome. So basically what it is, it just condenses and forms this X-shaped structure, which is usually what we see. So chromatin changes its form into a chromosome, and sometimes if it just condenses, it doesn't necessarily have to be. So these, like, it condenses first in this form, and then two of them combine together to form this. So both of them are called chromosomes. And so I don't want to confuse you, but since when it condenses, when the chromatin condenses, it is called chromosome. And when two chromosomes combine to form this structure, they're still called chromosomes. But when they're connected individually, they are called chromatids. And when they separate again, they're again, like the first part, they are still called chromosomes. So it's a little bit confusing, but this is the way they choose to name them. So anyways, so just a reminder, this is a chromatin. Like DNA and proteins, DNA chromatin condenses to form this structure called chromosome. When two chromosomes combine together, they're still called chromosomes. And each individual part, when they're connected, is called a chromatid. Anyways, the second part is called the second part, which I drew in the nucleus, is called the nucleolus. And the nucleolus is basically rich in RNA. So DNA and the other kind of molecule which we have is called the RNA, which is called ribonucleic acid, and that one is deoxyribonucleic acid. It's lacking an oxygen right there. So nucleolus is rich in RNA, and a special kind of RNA, actually. It's called ribosomal RNA, rRNA, which is used to manufacture ribosomes, which is right over here. And as we move on to ribosomes, I'll explain you which is basically an important organelle in the cell, which is present in both in eukaryote as well as in prokaryotes, because it manufactures, it basically is the point where the information of the nucleus goes to this organelle, and it produces protein which acts on specific parts of the cell, or even maybe outside of the cell if it needs to. So this is basically, it was in just, I hope you find it useful. It was basically just an introduction about the nucleus and about the different parts and the chromatin and the nucleolus. And the second part which I wanted to discuss was the endoplasmic reticulum. So basically endo means inside, just let me move this a little bit down. Space. Yes, so as I was saying, endo is basically means inside. Plasm means inside the cell or in the cell fluid, and reticulum means little net. So inside the cell, a little net. So you might be getting a picture of what endoplasmic reticulum is. So if I were to draw the nucleus, so let me draw the nucleus a little bit bigger so we are able to view it more clearly. If I were to draw the nucleus, let me draw the nucleus. So this is the nucleus. It's not the cell. It's basically the nucleus, which I'm drawing right here. And so this is the nucleus, and this is the nucleolus, and these are the chromatin, the genetic material in chromatin form. And so outside the nucleus, as I mentioned, there are nuclear pores, right? So, and if you observe in a microscope, you're able to see like a structure similar to this, like this. So this structure is called the endoplasmic reticulum, and what basically it does is, like, as a nucleus wants to send any kind of information to a part of the cell, it first goes under transcription, which is just that the strain of DNA, like if it's a double helix structure, it first, like, it unbounds itself and it makes a copy of itself, but the copy is not exactly the copy of the DNA, but it's the copies, it's I mean, it's the same, but the DNA is transcribed. The process is basically called transcription. It's transcribed into mRNA. Transcription. It's basically transcribed into mRNA, and I could do a video on this. So basically, in which we will discuss about different kinds of base pairs of the DNA. So, but I'm just giving you a general view that the DNA basically makes, from DNA we get an mRNA. So from DNA we go to mRNA, and from mRNA it's just basically a strand like this, like a piece of rope, you can view it as. And as it goes through the nuclear pore, it goes into the endoplasmic reticulum, and from endoplasmic reticulum it goes into the ribosomes to be translated, and then as it gets translated, it goes into so it basically, from mRNA it goes into the endoplasmic reticulum. Now it first goes into the ribosomes, and from the ribosomes it goes into the endoplasmic reticulum, and at that point it's basically a protein. And from the endoplasmic reticulum it goes into the Golgi bodies. So you might be thinking, okay, now I know why he selected these organelles, because they are basically interrelated with each other in some kind of sense. So we are right here. So the transcription is just an overview that where the DNA transcribes into mRNA, and mRNA strand leaves the nucleopores, like leaves the nucleus through the nuclear pores, and it goes into the endoplasmic reticulum. So now first I want to discuss about the endoplasmic reticulum right here. So the basically endoplasmic reticulum are of two types. One is called the rough endoplasmic reticulum, and the other one is called smooth endoplasmic reticulum. The rough and the smooth. So the smooth lacks nucleus. Sorry. The smooth lacks ribosomes. And you can now imagine, like, and you can guess now, right, that the rough doesn't. It has ribosomes in it. Has ribosomes. It has ribosomes in it. Now, basically, instead of besides, like, aside of providing a way for the mRNA to travel along and go to the Golgi bodies, the smooth endoplasmic reticulum has different kinds of functions in it. First of all, the smooth endoplasmic reticulum basically has, well, basically there are three functions of the smooth endoplasmic reticulum. Three functions. So one function is that it is basically related to producing hormones such as steroids. So it produces steroid, which is in the ovaries or in the testes of male and female. So these hormones are produced, and which are produced by the smooth endoplasmic reticulum. The second function of the smooth endoplasmic reticulum is that it detoxifies the effect of the drugs. So the second function the first would be produces hormones, and the second one is that it detoxifies the effect of drugs, harmful drugs actually. Detoxify the effect of drugs. And the third function is that it stores calcium ions. So you might be thinking, okay, what's the function of the calcium ion? So when as the nerve cell or your brain cells active your muscle cells, first of all these calcium ions are stored in the membrane of the smooth endoplasmic reticulum, and when the nerve cell stimulates a muscle cell, the calcium ions are released from the membranes of the smooth endoplasmic reticulum, and they go to the muscle cell to help it contract. So basically it has three functions. The smooth endoplasmic reticulum, it produces hormones, detoxifies the harmful effect of the drugs, and it stores calcium ions in it. That's the second part, the rough endoplasmic reticulum, which has ribosomes. So you might already, I told you what the function of the rough endoplasmic reticulum was, that when the mRNA passes through the ribosomes, it basically forms a polypeptide chain or a protein, and sometimes polypeptide. So let me write that down. Polypeptide chain, and sometimes. So a polypeptide chain or basically a protein, and sometimes a carbohydrate molecule or a sugar molecule comes and attaches to it when the polypeptide chain is within these compartments or within this space. So and then it turns and then it converts into a glycoprotein, which is basically glucose and protein. Glycoprotein. So as this so that there are also functions apart from that. Endoplasmic reticulum helps in producing, the endoplasmic reticulum also helps in producing the cell membrane, so the outer membrane of the cell, which is called the cell membrane. It also helps to produce the plasma membrane or the cell membrane, because it's the enzymes of the rough endoplasmic reticulum produces cell membrane too. And as we go to the Golgi bodies, so the next part, the first part, so a little bit of review here. So the smooth endoplasmic reticulum has three functions: steroid, detoxifies, and calcium ions. And the rough endoplasmic reticulum helps in producing the protein, and it basically produces the plasma membrane of the it basically produces the plasma because it has phospholipids. The enzymes has phospholipid. The enzyme helps to produce phospholipids, and the phospholipids is a part of the cell membrane too. So the rough endoplasmic reticulum basically has two functions: produces cell membrane and helps the protein to go to different stages, such as the polypeptide and the glycoproteins, and eventually it turns out when it leaves the endoplasmic reticulum, it's basically a vesicle, which is this protein inside a membrane is called a vesicle. And as this vesicle goes, it then the process is not complete yet until it goes to the Golgi bodies. This part right here. So just let me scroll down a little bit. So as it goes down from the endoplasmic reticulum, it goes towards the protein goes towards the Golgi bodies. Now, the Golgi bodies is basically, it's a little bit similar. So you might be wondering, why is he drawing the sides a little bit like this shape? Just need to remove this. Just a little bit. Oops, wrong. Yes. Now you might be wondering, why is it producing this? So basically the Golgi bodies has a receiving side. This is called the receiving side. And a shipping side, this part. So basically the function of the Golgi bodies is, as the protein leaves the endoplasmic reticulum and comes into Golgi body, it processes and it wraps it around. It's sort of like a packaging factory where it wraps it around a membrane and prepares it to go to any part of the cell and perform its function, which is encoded in the protein, which tells it what to do. And for a little bit of history for this organelle is that it was actually one of the first organelles to be discovered by Italian scientist named Camillo Golgi, and the Golgi is named after him. At first, nobody believed him, but when the electron microscope was invented, so this confirmed that the scientist Golgi was right. There are such organelles. And so they named this organelle in the honor of Camillo Golgi. And basically it's a manufacturing, it's a place where all the proteins come and they get wrapped around. It's like a package, and they are sent to different parts of the cells, even outside of the cell, to perform their functions. So, I almost forgot to tell you about the ribosomes. As I was saying, the rough endoplasmic reticulum has ribosomes, and the smooth endoplasmic reticulum doesn't have ribosomes. So basically we covered what ribosomes does, as the RNA. So ribosome basically has two parts: a larger unit, which is like this, and a smaller unit. And you might be wondering, so this is the endoplasmic reticulum right here, and the proteins which go through here and end up inside the membrane of the endoplasmic reticulum right over here. So as the mRNA goes through this, it is converted into a protein, which is just amino acids like this, which eventually goes through which eventually leaves the endoplasmic reticulum, goes to the Golgi bodies, does the packaging process, and it goes to different kinds of parts of the cell where it needs to perform different kinds of function. So in the next part, we are going to discuss about lysosomes and vacuoles, which are basically related to hydrolytic decomposition, and which are basically storage and decomposition process and decomposition. And hope you find this video useful. I'll see you in the next video. Bye bye.